So still considering those pea plants, we're going to go ahead and look at a dihybrid cross and how genes sort in this dihybrid cross. So now we're looking at two characters as opposed to just one character. We're going to look at seed shape and seed color. Now, the different traits associated with these characters can be seen above, whereas the seed shape could either be uppercase R, round, or lowercase r, wrinkled. Those are the alleles that we're going to look at for seed shape. Seed color can either be uppercase Y, yellow, or lowercase y, green. So Mendel went ahead and ran his experiments, got his P generation. Remember, that's the purebred parent generation. So here, purebred, round and yellow seeds. And here, the purebred recessive, that's going to be wrinkled and green seeds. And he crossed them. Cross-fertilization, bringing those alleles down, and considering that if these alleles were dependent on each other, what I mean by that is that the round allele would go with the yellow dominant allele. They'll both go together. And the same for the recessive. They depend on each other, so they're going to go together. Mendel would assume that the F1 generation would be this heterozygous individual where you have those different alleles, one dominant R, one recessive R, one dominant Y, one recessive Y. So round, wrinkled, yellow, green, showing those dominant traits because the dominant masks the recessive, showing round and showing yellow as seen in this image. So then Mendel would cross-fertilize F1 generations, our hybrids, if you will, and what would happen here if we ran our punnet? Yeah, so what would, just for showing you, if you'd run the punnet, then those alleles, if they are dependently assorted together, they come together, then we would see these different offsprings being shown. Running the punnet, we would see round and yellow, 3 out of 4, and we would see 1 out of 4, green and wrinkled. That doesn't actually happen. That's not what Mendel actually saw in this generation when he grew the peas. What he actually saw was the next scenario. So again, exact same thing seen here where we have our pure, round, yellow parent, purebred, green, wrinkled parent coming together to form our F1 hybrid heterozygous individual showing both dominant and recessive alleles. What Mendel actually saw in the F2 generation with the mating of the F1 parents was this combination. And so we actually get a dihybrid cross showing here for two characters. And the reason why, I'm going to explain it further in a bit. The reason why is because alleles independently assort. They don't depend on other alleles. What I mean by that is that the round variant is not going to be dependent on the yellow variant. It doesn't care that that trait exists, yellow. It's just going to assort independently of the yellow. They just so happen to combine that way. What I mean by this is, check it out. So here we have phenotypes, yellow round, green wrinkled. So if we look at our dihybrid cross down here in our F2, I do see round yellow. There's lots of those offspring round yellow. In fact, we have 9 out of 16 being round and yellow. And then this parent right here is green and wrinkled. I also see that, green and wrinkled. That offspring does form. Only 1 out of 16 does. I actually see some offspring being formed here that didn't exist in previous generations. Here I have a round but green seed. In fact, I get 3 out of 16 showing those traits, round and green. Okay, that's interesting. What also didn't show before is I have yellow and wrinkled also existing. There's 3 out of 16. That appeared in the F2 generation. So what's going on here is that these alleles independently assort from each other. We're considering 4 alleles. Remember, round, wrinkled, yellow, and green. none allele is dependent on another allele. They're just going to assort different from each other, and the way that they assort gives us these different combinations. So in lecture, I just want you guys to appreciate that we don't get these results from a dihybrid cross. We don't actually see that 3-to-1 phenotypic ratio. We actually get a 9-to-3 to 3-to-1 phenotypic ratio. So to further show you guys, this is Mendel's law of independent assortment. That is, that a dihybrid cross is going to result in a 9-to-3 to 3-to-1 phenotypic ratio.